Hello, everyone. It's my great pleasure to introduce Dr. Hesam Jahanian, who is an assistant professor at our UW Radiology department. Dr. Jahanian received his PhD from University of Michigan, Ann Arbor, where he worked on developing techniques for imaging blood flow in the brain using MRI. And then he did a postdoctoral training at Stanford, where he worked on developing ultra-fast MRI imaging techniques. Now, here at UW, his lab focuses on combining AI with MRI towards early detection of neurological disorders, such as Alzheimer's disease. Without further ado, I hand it over to Dr. Jahanian.
Thank you very much, Dr. Yazdan, for the introduction. It's a great pleasure to be here and talk to you guys about MRI-based methods for early detection of Alzheimer's disease. If you have any question at any point, feel free to raise your hand. I will be happy to answer your questions. In this talk, first, I'm going to start by talking about Alzheimer's disease itself and make sure that we are on the same page about the underlying mechanism and pathology of Alzheimer's disease. And then I'll talk about the importance of having biomarkers for Alzheimer's disease specifically. And we will talk about the current biomarkers that we have for AD. And finally, I will move on and explain some of the ongoing works that we are doing at Alzheimer's Disease Research Center and Integrated Brain Imaging Center at University of Washington to improve the biomarkers that we have for Alzheimer's disease. So Alzheimer's disease, it's a progressive neurological disorder that causes the brain to shrink and neurons to die. And it typically begins with subtle memory loss, but eventually cause continuous decline in cognitive ability, memory, thinking, and eventually impairs independent function of the patient, unfortunately. It's the most common cause of dementia. Currently, approximately 1 in 9 adults above 65 are dealing with Alzheimer's disease. And it's important to differentiate dementia from Alzheimer's disease because there are other kind of diseases that could cause dementia, but Alzheimer's disease is the most common cause of it. And it is defined by characteristic brain pathology that we're going to talk about in the next slide here. AD is characterized by formation of 2 abnormal proteins. The first one is beta amyloid. Beta amyloid is a protein fragment snipped from an amyloid precursor protein, which has an important role in synaptic formation and repair. So in healthy brain, these protein fragments are broken down and eliminated and cleared out of the brain. But in Alzheimer's disease patients, these fragments accumulate and form a hard, insoluble plaques between nerve cells. And they are called beta amyloid plaques, these dots, black dots that you can see here. So this is the first abnormal protein that, in Alzheimer's disease. The second one is tau protein. Tau basically binds to microtubules inside the neurons and assists with their formation and stabilization. But abnormal phosphorylation of tau can lead to deposit of tau filaments, causing them to aggregate in an, again, insoluble form inside neurons, which is called neurofibrillary tangles. And the combination of these two eventually lead to neurodegeneration and death of the neurons. So there are 3 steps: these formation of these abnormal proteins, and then neurodegeneration that leads to the cognitive problem that Alzheimer's disease patients have. The thing that is makes Alzheimer's disease very challenging to deal with is the time course of the disease. So as I mentioned, it is believed to be started, the disease believed to be started by formation of abnormal amyloid beta and tau protein. The thing is that the disease has a very long preclinical period. So after start of formation of these proteins, for decades, typically more than 20 years, the patients are cognitively normal. There is no symptoms. And only after many, many years is when we start to see some symptoms, and they are typically subtle memory problem. And after that, the patient enters a phase which is called mild cognitive impairment. And this is where we start to see neurodegeneration happening in the brain, and the symptoms progressively get worse. And after a few years, many of these patients develop full dementia and get completely impaired in terms of cognitive ability. And in clinical setting, this is currently the point that we diagnose the disease. The problem is that at this point, it's too late, and the irreversible damage has already been done to the brain. And no matter what treatment or drug we use here, it's almost impossible to reverse the damage that has been done to the brain. And this is actually believed to be one of the main reasons that the clinical trials for Alzheimer's disease has so far mainly failed. To be able to have the highest chance of success for any treatment or drug, we would like to apply them in this area at the earliest stage of the disease, before all those neurodegeneration completely happening. The problem is that at this stage there is no cognitive, there is no symptoms, and the patient is cognitively normal. So it's hard to identify those patients. To be able to identify them, we need to rely on some techniques to measure some physiological parameters that are related to formation of these two proteins. And they are called biomarkers. So because of this long preclinical period, having a biomarker is very important and central in Alzheimer's disease research. If we have a very sensitive and specific biomarker, it can be very useful in AD research and also in clinical setting, because as I mentioned, it can help us with early diagnosis of the disease, but it can also improve our diagnostic certainty, because as I mentioned, dementia can be caused by different diseases, and without these biomarkers, it's really hard to pinpoint the exact disease that is causing the dementia. We can stage the disease, monitor the progression of the disease, and also in the future, we can even predict the trajectory of the disease in each individual, and basically personalize our treatment or medicine for each individual. And it can also use to predict the response to a treatment. And it is very important and can accelerate the development of drugs in Alzheimer's disease, because in human trial of Alzheimer's disease drug, we cannot apply, test the drug and wait 20 years to see what happens. So these biomarkers here can help us to basically evaluate the effect of the drug on a very short period of time and accelerate the development of it. Fortunately, currently we have some biomarkers for Alzheimer's disease. Here I'm going to talk about 3 of them. The first one are methods that are based on analysis of cerebrospinal fluid, or CSF. The thing about CSF is basically a liquid that surrounds the brain and goes into the spinal cord, and it provides us with a window into what's happening in the brain in terms of like metabolism or the pathophysiology of what's happening in the brain. So in these methods, by doing a spinal tap, we extract some of the CSF from spinal cord, and by analyzing them and measuring the level of A beta and tau protein in them, we can have an estimate of what's happening in the brain without actually reaching to the brain. So this is the first category of Alzheimer's disease biomarkers. The second one is based on using positron emission tomography, or PET. PET is a biomedical imaging modality that is very versatile. It's very specific and important because using PET, we can image the concentration of a specific molecule in the brain. So it is also called molecular imaging. For any PET experiment, first we need to develop something called a PET tracer. PET tracer is simply a chemical molecule of interest combined with the positron emitting isotope, which is a radioactive material. That radioactive material basically just labels that molecule that we are interested in. After developing this PET tracer, we inject it into the bloodstream, wait for it to travel through the body, and wait for the chemical molecule of interest to bind to the molecule that we are interested in. After that, we wait for a while, and this positron-emitting isotope will decay and emit gamma rays in opposite direction that we detect using this ring of detectors in the PET scanner. And by doing some reconstruction and some processing, we can create an image which intensity corresponds to concentration of that radioactive material in that particular location in the body. And because that radioactive material is bound to the chemical molecule, basically it would give us an estimate of the concentration of that molecule in the brain. In case of Alzheimer's disease, by using different PET tracers, we can use a chemical molecule that would bind to tau and create an image of the concentration of tau protein in the brain. And we can do the same for beta amyloid plaque. We also can measure the neurodegeneration by using a chemical molecule that is similar to glucose, and it would enter the glucose metabolism cycle and get trapped in there. And because of that, the concentration of that molecule would be corresponding to the rate of metabolism. And when neurodegeneration happens, the first step is the decline in metabolism. So for example, here you can see in this area, the rate of metabolism has been declined compared to other parts of the brain. So it shows that there is some neurodegeneration happening here. The good thing about it is that it's more sensitive, and we can detect neurodegeneration at early stages. Another biomarker that we can use for AD is using MRI, and particularly structural MRI. We can use it to basically image neurodegeneration. With MRI, we can create high spatial resolution anatomical image of the brain, and by measuring like the cortical thickness in areas like hippocampus and entorhinal cortex that we know are particularly affected in AD, we can estimate the neurodegeneration. So these are the most common techniques that we use in clinical settings for AD. Each of these methods has some advantages and disadvantages. CSF and PET biomarkers, they're reliable, sensitive, and provide a specific diagnosis for AD. But both of them are invasive with potential side effects. And more importantly, they're expensive, and in terms of availability, they're very limited, particularly the PET imaging. They're very expensive and not available at all clinical settings. And because of that, they cannot be used for routine clinical screening. And remember, it is important because we need to apply it on cognitively normal people as a screening tool. So basically, we need to screen every older adult above 65 every year. And because of that, it is important for the method to be non-invasive and accessible. In terms of structural MRI, it's easy to acquire, no radiation, and because of that, it can be repeated over time to monitor the progress of the disease, but it's not sensitive to early stages of the disease. And by the time that we are seeing some atrophy in the brain, we are way past the mild cognitive impairment period. And because of lack of sensitivity, it's not very helpful in our mission to detect the disease at early stages. And for these reasons that I mentioned, currently there is a pressing need for an accurate, sensitive, specific, widely available, and non-invasive biomarker for AD. And for the rest of the talk, I'm going to talk about the alternative approaches that we are pursuing here at University of Washington. We are interested in using MR-based methods to develop biomarkers. There are two different approaches that we are basically following. The first one is resting state fMRI as a biomarker, using resting state fMRI as a biomarker. And the other one is synthesizing PET images from MRI data using deep learning. So let's talk about each of these methods. First, let's talk about the resting state fMRI itself. It's a category of functional MRI methods. Functional MRI is one of the very popular methods in functional imaging. Basically, in medical imaging, we have two categories of techniques: structural imaging, where we create like an image of the anatomy of the patient, and the physiological information is interpreted. So for example, as I mentioned, we have a structural image. We see atrophy in hippocampus area. We can conclude that the patient has some memory problems. But in functional imaging, we go and directly measure some physiological information. So for example, with PET, we go and directly measure the concentration of tau, A beta, or neurodegeneration. And that's a form of functional imaging. And functional MRI is the technique that we use MRI to basically indirectly measure the neural activity in the brain. Resting state fMRI is like a specific flavor of functional MRI in which the subject doesn't perform a task. So in a resting state fMRI experiment, the subject lays in the scanner at rest for an extended period of time, typically about 10 minutes. And we continuously image the patient during that time. And then we follow the changes in signal intensity at each voxel of the brain. We call it time course. So for example, we extract time course from one area that we are interested in, here like from PCC, and then compare the time course of other areas of the brain with that reference time course and identify the ones that are highly correlated. So this highly correlated area forms something which we call functional connectivity networks. And they are believed to areas of the brain that are connected to each other and gets activated and deactivated together. Many functional connectivity networks has been identified, and they are shown to be related to different disease or different tasks. In case of Alzheimer's disease, this functional connectivity network shown here, called default mode network, is shown to be altered as a result of Alzheimer's disease. It has been shown that the connectivity in the hippocampal and entorhinal cortex has been decreased as a result of AD. So it's promising. It tells us that it is sensitive to changes that happens because of AD. And again, it's like any MRI. It's easy to acquire, no radiation, test can be repeated, and patient can be evaluated because unlike task-based fMRI, the subject doesn't need to perform any task in resting state fMRI. The problem is that the results are not reliably interpretable at the single subject level, mainly because our data is too noisy. And because of that, the contrast to noise ratio is low. So at single subject level, we cannot basically do the measurements reliably. So what can we do to improve this contrast to noise ratio and make our data less noisy? People have approached this problem with different solutions by improving the acquisition, by improving the pre-processing, meaning that by improving the way that we are denoising the data, and also by improving the processing, the way that we are analyzing the data. What can we do to extract more information and more useful information from the data? So I'm going to talk about these two a little bit. So the first approach is by accelerating the image acquisition. Conventionally, for fMRI, the images are collected sequentially, one slice at a time. And because of that, it takes a long time. It takes more than 2 seconds just to image one volume of the brain. So the temporal resolution is pretty low. What we can do with ultra-fast fMRI is collect multiple slices at the same time. And because of that, we can improve the temporal resolution significantly. So for example, now we can image one volume of the brain in about 300, 350 milliseconds. It seems to be like a very simple solution, but it's actually more complicated than that, because when we are imaging more than one slice at a time, if we just reconstruct the image using the traditional methods, we will end up with an image of 2 slices on top of each other, and we need to do some advanced parallel imaging and reconstruction to separate them from each other. But by improving the temporal resolution, first of all, we have more samples, which will increase the statistical power of our measurement, but it also increases the bandwidth of the signal that we are sampling. One of the main problems in resting state fMRI is the physiological noise, meaning that the physiological noise is any physiological phenomenon in the brain that modifies the fMRI signal that we are measuring, that are not due to neural activity. So they are confounding our measurements, and we would like to remove them. When our temporal resolution is low and it's very poor, these components get aliased into the signal band and combine with them, and they are very hard to remove. But when we have improved temporal resolution, we can simply filter them out. And also we could include higher frequency component in the data. By combining these two, we can improve our pre-processing. Here I'm going to show you an example. The same data, both of them are high temporal resolution data. On the left, we pre-processed it using the traditional method, and on the right, using our optimized method. And as you can see, for the optimized method, in about 30 seconds, the maps get to a steady state and we have a clean map, whereas for the conventional pre-processing method, it takes more than 2 minutes to get a clean map, similar to what we had from 30 seconds of optimized method. And by doing so, we showed that we can improve the sensitivity and also reliability of our measurements. So from a shorter period of time, we can have better quality maps of default mode network. So what we can do with that? Traditionally, from 10 minutes of a scan that we talked about, the output is one stationary map of default mode network. But now that we can measure the map from a shorter time, we can look at the dynamics. By measuring just one stationary map, basically we have assumed that the default mode network is a stationary process and it's constant during the scan. But we know that from electrical physiology that it's not a valid assumption. But by doing the dynamic imaging, now we can see and measure the dynamic of the default mode network in the brain. We can see how dynamic this system is, how different nodes get connected and disconnected to the network. And there is a wealth of information here that before we could not actually use and improve the sensitivity of our measurements. It's like comparing a picture to a movie. And now, with these advances that I mentioned, we are able to look into dynamics. So we use different methods and develop different methods for dynamic resting state fMRI measurements. One method is simply estimating the default mode network over a short period of time, and then slide that window and do the measurements again. It's called a sliding window. And by doing so, we can have multiple maps of default mode network, and using k-means clustering, we can group them into different states. So basically, we assume that default mode networks switch between different states during the rest. And for each point, we assigned each of these maps to one of these states. And by looking at the transition between these states for each subject and extracting some features from them, we can then differentiate between AD and control. So for example, we saw that the percentage of time spent in state 3 can be a good indicator and used to be classified AD from healthy controls with accuracy of about 80%. And given the limited number of subjects that we had, was very promising. Another approach that we took was using the DCC Garch model, which basically modeled the correlation between time courses. So I'm going to show you what the model does using this example. If you just compare the time courses from 3 nodes, these 4 time courses, by just looking at the time course, you don't see any significant difference between them. But when you look at the pairwise correlation between these time courses, you see that these the pattern of correlation is significantly different between them. And by using those parameters alpha and beta, which basically models how fast correlation changes and how volatile this correlation can be, we can differentiate these different patterns of correlation. And we saw that by just looking at the DCC alpha parameter, we can separate AD from non-AD patients, whereas by just looking at the stationary functional connectivity in default mode network, we could not do that. Okay, so I'm going to talk about the second approach, which synthesizing PET images from MRI data using deep learning. And in this approach, sorry, how much time do you have? Any questions so far? Yep.
I just wanted to ask about, how does this work with the notion of genetic testing to identify at-risk patients early?
Mm-hmm.
And since CSF measures chemical changes in your spinal fluid, would that be corresponding to changes in, say, different chemicals in your blood analysis? And if so, would it be possible to create a diagnostic tool from just a blood draw?
Yeah, that's a good question. First of all, there are some genes that has been identified and shown to increase the chance of getting the Alzheimer's disease. And we are using those to basically identify or cohort for clinical studies and in research. But for clinical setting, because it just increased the chances of getting the disease by about 50%. It's not definite. So again, we would still need to have biomarkers. Without that, we cannot just by looking at the genetic material conclude that this person will definitely have Alzheimer, and for example, we need to start a treatment or start taking a drug just based on that. And in terms of analyzing the plasma from a blood sample, that's actually one of the active areas that people are working on to be able to detect those materials from blood samples. The problem is that in brain, we have something called blood-brain barrier, which makes it very hard for these proteins to pass from brain to the blood. And some of them pass, but it makes it much harder. And because of that, the concentration of those proteins in the blood is much lower compared to CSF. And that makes the measurement much harder, much noisier, less reliable. Again, it's one of the active areas that people are working on, and a great progress has been achieved in that area as well. But still, we are not there. But that's one of the very promising line of research to develop very accessible biomarkers for Alzheimer's disease. Any other questions? Okay. So we're going to move on to the second approach, which is synthesizing PET images from MRI using deep learning. So in this approach, basically our hypothesis was, so the good thing about MRI is that it is not just one form of image. You can collect many forms of image from MRI with different contrast. It's called different contrast. It can be anatomical images with different contrast. It could be images related to the blood flow. It could be images related to the concentration of bundles in the brain. It could be images related to functional. So there's a vast range of physiological parameter that we could information that we could get from MRI. And our hypothesis was that whether the information that we get from PET can be reconstructed and extracted from the multi-contrast MRI data that we are getting. So basically, we were looking for a function that could map the multi-contrast MRI data to a PET data and reconstruct an image from that. Specifically, we started by looking at this contrast, T1 contrast, flare, and ASL, that I'm going to talk about them later. And we would like to reconstruct FDG PET, which is those images that I show you that was related to rate of metabolism, and we were using them to detect neurodegeneration at early stages. So to be able to do that, the input images were T1 and T1 contrast. There are two kinds of structural image used with MRI. The difference is that in the second one, we inject the subject with a gadolinium contrast agent. Flare is, again, another structural image in which the CSF signal has been nulled. And ASL is the kind of contrast and MRI image that we use to measure the blood flow in the brain. The reason that we use this combination is that ASL basically measures the blood flow in the brain, and we know that the blood flow is related to metabolism. And because of that, we thought there's a logical path from ASL to PET, and we should be able to extract the same information from MRI as FDG PET. So in ASL, the way that we measure the blood flow is that we first collect two sets of data, the control, which is like a typical MRI data, and the label one, or tag image, in which we magnetically label the signal that comes from the blood, these pink arrows here. And because of that, when we subtract them from each other, we will end up with an image that its intensity is related to the blood flow. So the setup of our experiment was like that. We have our input MRI multi-contrast data. We looked at the different patches of the data to basically augment the data. And then we train a deep convolutional neural network that could estimate an image similar to the ground truth PET. So we would iteratively compare the output of the model to the ground truth and update the parameters in the model until we get to the steady state, and it is called training of the model. And once the model was trained, then we could use any set of input multi-contrast data and get like a synthesized FDG PET from that. So, and the architecture that we used was unit architecture, which is shown to be very effective in segmentation of medical imaging data and also image reconstruction. It has an encoding and decoding path. During the encoding, as we go deeper and deeper into the network, we move from image space to the feature space. So here we have the most feature representation of the image. And then we go back to the image space, and there are some forward connection as well to pass the spatial information from input to output. And this is basically the standard unit. We also modified this model to make it perform better by specifically adding a split input model and the attention modules and the transfer layers. What the split input module was that basically we would put a weight for each of these contrasts. Because with the standard architecture, we are basically assume that the importance of each of these input contrasts is the same in generating the FDG PET. While we know that it's not exactly the case, and for example, ASL, turns out ASL and T2 flare were the most important ones. So by putting a weight here, we are basically telling the model to focus more on these inputs. And attention modules basically do a similar thing. Within like an image, we are basically telling the model that there are some areas that are more important than others. And here is the areas that have some pathology to them. So we are basically asking the model to pay more attention to some parts of the model. And because of that, it helps us a lot to basically preserve the pathology. So in the next slide, I'm going to show you a couple of examples of the results that we got. Yeah.
Just wondering, in the previous slide for the MRI, you showed that there is a time component to it. So I was just wondering, is there any reason why you're not using a RNN or a recursive kind of study for your model?
So for the ones that we used here, they were just three-dimensional data. We didn't include functional data on them. Yeah, so those T1 contrast, T2, and ASL, both of them are just 3D images. Yeah. So this is the first example. This is a real FDG PET image from a patient that has a brain tumor. The part that is being highlighted with the red rectangle here is the part that we have the pathology. If you compare it to the other side, you see that we have some abnormality here in the area that we have the tumor. And this is what we got from, this is the synthesized PET, what we got from by just extracting information from multi-contrast MRI data. So the most important thing is that the model preserved the pathology. So basically, if I show each of these images to a radiologist, the diagnosis that they would make would be the same. And that's the key point here. Here's another one. Again, we have the pathology here, and this is the output of the model. Again, very similar, and most importantly, it preserved the pathology. Here's another one. We have the tumor here. And this is what we got from the model. So overall, as you can see, it was very successful in creating the images that not only look a lot like the ground truth, but it is also preserving the pathology. And here we also compared our model to standard unit and attention unit models and show that it is performing better, both in terms of like the details of the image and also preserving the pathology. Here's again another one, another example. Again, in these two models, you can't really see the area that we have hypoperfusion and hypometabolism. Here's two other ones. Again, we have a similar pattern that using the standard architectures of deep neural network, we could not really preserve the pathology. And it is due to the attention modules that we added to the model. And for example, if you look at the spatial attention map that we get from the attention unit and compare it to proposed method, you can see that our model put a lot of attention to the area that we have the pathology, where attention unit was could not really detect that and put a lot of attention there. Same goes here. We have pathology here, and we see some attention going there, whereas in attention unit, we didn't have that. We also compared like our model to others by quantitatively comparing the results in terms of peak signal to noise ratio and structural similarity and root mean square error. And for all of them, the proposed method was performed the best. So hopefully I could show you here that dynamic functional connectivity provides a more sensitive, is more sensitive to AD pathology compared to traditional stationary functional connectivity measurements. And it's promising, meaning that it has the potential to be used as a biomarker for Alzheimer's disease. And also I showed that the deep learning model, using the deep learning model, we can generate high quality and clinically meaningful PET images, similar to what we get using a real PET without using the pathology and by preserving the details of the image. And lastly, I would like to acknowledge our collaborators, Dr. Zarchuk, Dr. Grabowski, and Dr. Shujai, and also the students that help us with this research. Particularly the deep learning project was in close collaboration with Stanford University and with collaboration with Dr. Zarchuk. And thank you very much. I would be happy to answer any question that you might have. And if you're interested in any of these projects and would like to chat more about them or are interested in working on them, feel free to send me an email. I would be happy to chat.
Thank you very much for a very exciting talk, Dr. Jahanian. I'm going to open it up for questions. Go ahead.
Yeah, I have a question. So for the PET image reconstruction task, was the deep net trained on data from multiple patients or separately deep nets trained on data for each patient individually? And how much data was there like?
How much data did we use?
Yeah.
So we had about, overall we had about 100 patients. And for those patients, we have multi-contrast MRI and gram truth PET. We used about 70 of them for training, 10 for evaluation, and 20 for testing. But we did a lot of data augmentation technique, like using the patch and manipulating like the dimension and stuff like that to increase the actual number of training sets. But in terms of patient, it was about 100. Yeah.
Any more questions? Well, I have a question kind of following up what Felix was asking. So ASL has some sort of anatomical information. So what was like T1 or T1 contrast or flare? What was it that they were adding to that that like you couldn't get from ASL? Like basically I was just asking like, did you only use ASL to see if you can reproduce?
Yeah, that's a very good question. The thing is that it seems that ASL is providing some physiological information about what's happening there. T1 and T1 particularly, T1, T2, and FLAIR, all of them create and provide some anatomical information because the spatial resolution of ASL is much lower than those two. And T2 particularly because it knows the CSF signal, it adds some information about abnormality and like the extent of tumor that we have there. Each of those basically are adding some information to the model. We also try to train the model by using only a number of subset of these images. The results were still good, but when we were removing any of these, overall the quality of or sensitivity would decrease a little bit. So it's sometimes hard to interpret exactly what's happening in the model, but my understanding of what's happening and what each of these contrasts and images bring to the table was like that. They are ASL, mostly information about metabolism. T1 contrast, anatomical information, and T2 FLAIR, some information about the anatomy and also the extent of the pathology to the model.
Okay, and so I think one of the things that I'm assuming, but I think it would be good if you tell our students, is that why it would be beneficial to do MRI rather than PET. Because like if you get the same information, like what is it? So I think it would be good for people to know.
Sure, yeah. As I mentioned, the problem with PET is, first of all, in terms of availability, it's very limited. It's much more expensive than also compared to MRI. So there are only very limited centers that could do PET imaging, and the cost is also very high. The insurance doesn't cover it, so at least for now. But MRI is much more available. You cannot find a hospital that doesn't have a few MRI machines. In terms of cost, it's much cheaper, and also there is no risk of radiation involved. MRI is completely safe. You can get a scan 10 times a day without any problem. So a combination of these reasons makes MRI a much more attractive choice compared to PET.
Okay, thank you. Any more questions? Yes.
Yeah.
So actually I want to talk a little bit about the noise and the conversion from an MRI to a PET scan. In the resting state MRI, we talked about how cardiovascular systems input noise, same with like breathing. So wondering what sources of noise do you have when you're trying in the system, and how are those like accounted for when you do the translation from the MRI images to a PET scan image?
Yeah, that's a very good question. There are any MRI image that you get, they could have some different kind of like noise in them. There are some standard noise that like thermal noise that you see on any like MRI contrast or any MRI image. On top of that, motion is a problem. Whenever the subject moves, it adds some artifacts to the data. These are like general things. But in terms of sensitivity to the artifact, the anatomical images are very stable in MRI. Because we scan for 5 to 7 minutes, for example, it can give us a very high spatial resolution, high quality image. And unless the subject moves a lot, the images are typically very robust, and there are many techniques to remove the noise or reduce the sensitivity to the noise. But the story for ASL is different, because the way that we generate the contrast is much more complicated. there could be some artifacts in terms of like the signal in tagging those and labeling the signal that comes from the blood. It's very challenging. A lot of times it's not perfect. And there's some variability there. And also the quality of the image, sometimes it could be very low. The thing that we did to basically make the model more generalizable and less sensitive to these artifacts, particularly from ASL, was that to include more and more data with different quality in the model. So when we train the model using both low quality, noisy data and also high quality data, it makes it more robust to those sort of artifacts and imperfection. So in short, the solution is in adding more training data with different quality as much as you can. As long as you add them in and train the model using those data, it wouldn't be a problem.
Do we have any more questions? I guess I have one last question.
Sure.
And so I understand that, understanding these, and trying to help with the early detection of Alzheimer's disease is at its early stages. But where in that 20 years period you think these techniques can help? Like, is it like at the very early stages, or is it like how many years should pass from the early onset for these techniques to be able to detect?
So it depends. So the thing is that with the limited data that we have, so to be able to answer that question, we should continuously do these measurements on people that are cognitively normal and follow them over many, many years so that we could see at what point we can see basically and detect the accumulation of pathology. And the thing is that without biopsy or basically looking at the brain itself, it's very hard to say whether what we are basically measuring is the start of it or not. The hope is to be able to detect it as close as possible, but I don't think that even if we could detect it after 5 years from the start, that would be a huge gain. So at this point, we don't know. And we are collecting data, different institutions are collecting data to be able to answer that question and have a better understanding of the evolution of each of these biomarkers over time. But the more sensitive the method is, the higher the chances of detecting the disease as soon as possible.
Okay. Thank you so much. Let's give our speaker another round of applause, and thank you all for coming.
Thank you.